Get started. Good morning, everyone. Welcome to another Terasaki seminar series. Today we have Professor Phillip Messersmith from UC Berkeley. He's a class of 1941 professor in the departments of bioengineering and material science and engineering at UC Berkeley. He earned his bachelor's degree from University of Illinois at Urbana, master's in bioengineering from Clemson, his PhD in material science and engineering from the University of Illinois at Urbana. He was a postdoctoral fellow at Cornell University, faculty member of University of Illinois at Chicago and Northwestern University. He's published more than 220 papers and has 44 patents under his name. He's received many awards and recollections. These include the Merit Award from NIH, the Langmuir Lecture Award from American Chemical Society, and 2013 Clemson Award for Basic Research from the Society of Biomaterials. In addition, he's a fellow of the American Institute for Medical and Biological Engineering, the Royal Society of Chemistry, and International Union of Societies of Biomaterial Science and Engineering. The floor is yours.
Thank you very much. Thank you to the organizers for the kind invitation. I hope you can hear me. If not, send me a message or speak up. Yes, I'm pleased to participate today. I wish I could be there in person, but it's not in the cards, so I'm happy to be here remotely. So I want to talk about my work on supramolecular polymers for regeneration of tissues using a drug-induced strategy for regeneration. I should point out my collaborator on almost everything I'll tell you about today is Ellen Heber-Katz at Lankenau Institute for Medical Research in Pennsylvania. Just get the disclaimer out of the way. Ellen and I have formed a company to commercialize the technology that I'm going to tell you about today. So that's that. And so here's the outline of my talk. I'll start by basically taking a very historical approach, even mythological approach to regeneration in primitive organisms and adult mammals, and talk about an interesting model for tissue regeneration, which is the MRL mouse, HIF-1 alpha and the role of HIF-1 alpha, the transcription factor in regeneration, prolyl hydroxylase inhibitors to regulate HIF-1 alpha expression levels, and then regeneration in a couple of different systems, tissues. So I'd like to start with a little bit of Greek mythology here. You may know the story of Prometheus, one of the Greek titans who was punished by the gods for giving the gift of fire to humans. And this picture that you see here, this painting, is basically a representation of his punishment by the gods, which was that he was chained to some mountain, and every day an eagle would come and eat his liver. Okay. And because the liver regenerates, the eagle came back every day, and this was his fate. His punishment was he had to suffer that experience every single day. Okay. As I said, actually, it's interesting story because liver is actually one of the few organs in adult humans that does have the ability to regenerate, although I would say not overnight. It does have much more limited regeneration capacity. There's a few other examples in vertebrates, adult mammals, like antler regrowth and rabbit ears, where you see regeneration. But for the most part, in most adult mammals, regeneration is really not a significant observation. It's usually healing by scar formation. And so, of course, the whole field of regenerative medicine and tissue engineering has the goal of being able to do this, and so that's one of our goals as well. We're taking a different approach than other people that are taking interested in regeneration. We do not use scaffolds like the classic sort of approach to tissue engineering, nor do we use biologics. No growth factors, no cells implanted. We're doing this through activation of a pathway, a primitive regeneration pathway through a drug. Let me talk about epimorphic regeneration. So tissue regeneration in primitive organisms is sometimes referred to as epimorphic regeneration. So, in very simple organisms like the metazoans and also in insects, you have, the ability to regenerate large fractions of the organism when they're cut off. There are a couple examples in invertebrates. You may have heard about these, lizards and newts. Actually, you can cut off the tail of a lizard and a limb of a newt, and it regenerates. And so the lower left of this slide actually shows this process in a newt, where if you sever the limb, either here on the left or on the right there, and then wait about 70 days, you get a complete regeneration of a functional replica of the original tissue. And this has been lost during evolution in humans. So we do not have this ability. And so this is called epimorphic regeneration, like that seen in the newt, and it has a few characteristics. One is that after the wound, there's very rapid re-epithelialization of the wound, followed by a blastoma formation, which is a cluster of initially undifferentiated cells. There's evidence that cells dedifferentiate, then they divide, redifferentiate, and then form multiple tissue types. And so these are two hallmarks of epimorphic regeneration. The MRL mouse, which I mentioned a few minutes ago, is an interesting example in adult mammals where it's a rare observation that the organism has the ability to actually regenerate tissues without the formation of scar. And so this is a mouse. It's not a knockout mouse. It's a inbred mouse. It was originally developed decades ago for immunology studies and obesity studies. But my colleague Ellen, back almost 25 years ago, made observations that in her animal experiments, where they nicked the ears of the mouse to identify the mice, she noticed that they had, she would come back in a few weeks and the markings would be totally regenerated. And so what emerged from that observation was basically several decades of research in her lab to try and uncover the underpinnings of this observation. So this is one of her initial experiments, this plot lower left, which shows the closure of an ear hole. So the experiment is shown here on the upper right, which is at day 0. She punches a 2 millimeter hole in the ear through and through, and then measures closure and regeneration within that opening. And in a normal mouse, there's a little bit of closure, but it does not fully close. And what does happen here is essentially scar formation. Okay. But in the MRL mouse, you get complete ear hole closure. And in fact, after about 100 days, you get almost full functional regeneration of tissue. So you can see after 30 days, there's complete closure in the MRL mouse. And then in the interior of the tissue where there would normally be cartilage, eventually the cartilage comes back and forms fully functional tissue. You also get regeneration of hair in this defect. So the MRL exhibits many of the key features of epimorphic regeneration, regeneration without scar, interesting metabolic state of this organism, which is anaerobic glycolysis rather than oxidative phosphorylation. It does rapidly epithelialize its wounds and forms an early blastoma by day 2 after wounding. Cells appear to re-enter the cell cycle and then proliferate and differentiate into various tissues. And this is all sort of autonomous. So it's sort of morphogenetic autonomy, which is similar to what is observed in the newt, for example. So we started working together about 15 years ago, and this was from our first paper a few years back. And so we identified, I should say, Ellen identified HIF1 alpha as a main feature underlying the regeneration mechanism in the MRL mouse. HIF1 alpha is a transcription factor. I'll tell you a little bit more about that later. But this is a key experiment. So she generated HIF1 alpha luciferase reporter mice by crossing MRL with luciferase reporter mice, B6 cross or MRL crosses. And so basically these images that you see here, the two on the left at day 0 and the two on the right at 7 days after this ear hole punch experiment, show heat maps. And so the more intense colors here represent high levels of HIF1 alpha. First thing you can notice is that before the at the time of the wounding, you see much higher overall HIF1 alpha levels in the MRL mouse compared to the normal laboratory mouse. After 7 days, you see a little bit higher levels of HIF1 alpha in the B6 cross and very, very intensely high levels of HIF1 alpha in the MRL cross, including locations in the ear hole punch where the wound was created. So this was the basis of our focus subsequently on HIF1 alpha and ultimately developing materials and drug delivery systems to target HIF1 alpha in regeneration. A little bit more about focusing now on regeneration, what's happening after this, ear hole punch experiment, what happens in the actual defect. So the tissue images that I'll show are edges of the tissue defect. So we punch out 2 millimeter hole at day 0 and then follow this and take samples, and the tissue sections are cut along this line here. So it's basically a donut. We're slicing it in half, and so in the images, the defect where we've removed the tissue is above, and then the unharmed tissue below. Okay, so these are experiments here. Shows the immunohistochemistry for HIF1 alpha. So everything green here is HIF1 alpha, and you can see very, very high levels of HIF1 alpha in the defect early after the wounding. So also observe this by Western blot. In fact, if you look at the progression of HIF1 alpha with time after injury, it's an interesting progression actually. In the MRL mouse, the levels of HIF1 alpha, as I've already shown you, go very, very high within a week. After about 2 weeks, they attenuate back down to lower levels, although still higher than the B6 mouse. Okay, and I'll come back to this later because this is a progression of expression of HIF1 alpha that we would like to replicate in our drug delivery experiments by a therapeutic agonist for HIF1 alpha to sort of reproduce this sort of temporal scheme. We know HIF1 alpha is key to this process, and this is an experiment that shows this. So we surveyed a few siRNAs against HIF1 alpha. This one, siRNA 3 here, we identified as particularly effective. And so we administered this siRNA after wounding the mice in the way I described already, and then following ear hole closure, and then also looking through the luciferase reporters at HIF1 alpha levels. And what you can see here is that the animals administered the siRNA, their response, their regenerative response is impeded. Okay, so you don't get the closure that you observe in the MRL mouse. And so this seems to be very strong evidence pointing to HIF1 alpha as a key player in this regenerative response. I promised to talk a little bit more about HIF1 alpha. HIF1 alpha is constitutively expressed within the cell, so it's produced constantly. And the upper half of this slide describes the normal oxygen situation in a cell. Basically this pathway above shows the constant degradation of HIF-1 alpha. So HIF stands for hypoxia inducible factor. So this is normally a transcription factor that is in response to low oxygen levels. And so under normal oxygen levels, it's usually suppressed through this pathway. And the pathway consists of, among others, a key enzyme here, prolyl hydroxylase enzyme. It's actually a family of these that hydroxylate two prolines on the HIF-1 alpha protein. Okay, these two prolines that you see here. So HIF prolyl hydroxylase labels these prolines with hydroxides. This tags the proteins for destruction through the E3 ubiquitin ligase complex and proteasome pathway. So this is constantly happening. So under normal oxygen conditions, HIF-1 alpha is produced, but constantly held down. And you can see that oxygen is a cofactor with prolyl hydroxylase, which means that under hypoxia conditions, this pathway above is suppressed. And then you get high levels of HIF-1 alpha. HIF-1 alpha can get into the nucleus where it can exert its effects as a transcription factor and influence downstream gene activation. And it's quite complicated downstream. So there are at least 100 genes that are connected somehow to HIF-1 alpha. So the downstream story is very, very complicated. But this slide also indicates a therapeutic approach and a number of therapeutic approaches. Ours is to inhibit PHD. So if we can inhibit, even under normal oxygen conditions, inhibit PHDs from degrading HIF-1 alpha, we can keep HIF-1 alpha levels high. So that's our approach. There are a number of PHD inhibitors. So there are several of these approved. They fall into a number of different classes, 2-oxoglutarate mimics, iron chelators, active site blockers, etc. Okay, the PHD2 enzyme has an active iron site here. So that's believed to be how the iron chelators work. They bind the iron competitively. And so the drug that we have identified is not approved or on this list. It's an experimental drug. I'll just refer to this as 1,4-DPCA or DPCA for short. You can see the drug structure here. This drug is through this site right here, very good at binding metals. So we don't know for sure, but it probably would fall into the class of iron chelators. Okay, and in fact, we have an initial experiment shows that there's a chromophore here change that indicates in the presence of Fe2, you get significant chromophoric effects, indicating probably that the binding is in this site through the unpaired electrons on the nitrogen. But otherwise, this is a highly aromatic compound. And despite the nitrogens and the oxygens here, this is very hydrophobic. So it's a very, very poorly soluble drug. So our strategy is to transiently stabilize HIF-1 alpha by delivering this therapeutic, which is actually an PHD inhibitor, and therefore stabilize HIF-1 alpha. And do this through an injectable, biocompatible, and high drug loading system for drug delivery. Our first publication in this area took advantage of a two-component hydrogel, two reactive polymers dissolved in water. So these are basically two 8-arm polyethylene glycols with different functional group termini at the end of each arms. One of these has a NHS ester, and then the other is an N-terminal cysteine. When you mix these two together under neutral pH conditions, you get a native chemical ligation type reaction, which we showed earlier in this paper, which cross-links covalently this into a rigid hydrogel. So within a few tens of seconds of mixing two solutions, aqueous solutions of these polymers, you get a really nice hydrogel. So it was new chemistry that we reported in this paper. For 1,4-DPCA and regeneration, our first attempt took advantage of simply the formation of this hydrogel and this chemistry that I just described in the presence of crystals of that drug that I showed. Okay, and so that drug, as I mentioned, is very poorly soluble in water. So we form the hydrogels in the presence of drug crystals, and therefore entrap them within the gel. And then this is a in vitro drug release profile of the release of 1,4-DPCA from that native chemical ligation hydrogel. And you can see the gradual clearing from outside in of the gel. And within about 5-6 days, all of the drug has been depleted from the hydrogel and released into the surroundings. On this slide, I'm showing you in vitro immunohistochemistry data, showing you the upregulation of HIF-1 alpha produced in normal B6 fibroblasts. So these are fibroblasts isolated from normal B6 mice. You can see that when you expose these fibroblasts to the gel carrier without the drug, you get a slight upregulation of HIF-1 alpha and very, very prominent upregulation of HIF-1 alpha in the presence of the drug entrapped within the gel. Here's Western blot, evidence of that as well. And also we find HIF-1 alpha target gene activation. I mentioned that the downstream activation pathway is very complicated, but we see some of the expected gene activations for high levels of HIF-1 alpha, like for example VEGF. So these are both pro-angiogenic genes and pro-glycolytic genes that you see here. And so in vitro, this looks very promising for upregulation of HIF-1 alpha. Now to the in vivo experiments, and I would like to start at the upper left and point out that that gel that I described, we designed, or my lab designed for this purpose to actually be injected into the ear hole punches directly into the defect. That's a very common thing in biomaterial science. And the problem was that our initial animal experiments failed because the animals actually poked the gels right out of the holes. So their claws have perfectly fit into that 2-millimeter hole and poked it out. So Ellen said, "Well, Phil, I'm just gonna inject subcutaneously, like in the back." And I said, "Ellen, you're crazy. It's not gonna work." Actually, it did work, and the rest of the story utilizes basically injection of this hydrogel with the drug into the upper back in 100 microliter or 50 microliter injections, subcutaneous, 3 times. So this is the injection, the administration scheme. At day 0, where we create the 2-millimeter punch, day 5 and day 10. Okay, and this, based upon our in vitro data, will give us this roughly 2-week progression of upregulation of HIF-1 alpha, at least we surmise. Okay. Here's in the middle of this slide are the ear hole closure data. Okay, so the gel without the drug is shown in blue. We get a little bit of closure. This is due to scar formation. And then the gel with the active therapeutic in it basically shows the same behavior as the MRL mouse, which is after roughly 1 to 2 months, nearly complete or complete closure, depending on the animal, of the ear hole. And so you can see the tissue cross sections here. This is simple H&E staining showing the ear hole closure in one of the animals with the administered the drug. Again, we did the siRNA experiment showing that that HIF is key to the regeneration. So the animals receiving the drug plus siRNA against HIF had a reduced regeneration. And also the immunohistochemistry of the actual wound site shows very similar evidence of HIF1 alpha upregulation, as I indicated a little bit earlier. The other tissues, so the ear hole closure measurement is just basically a physical measurement. The dermis, epidermis in these animals, of course, fully close eventually, but also other tissues. Of course, none of this can happen without vascularity, and nothing on this slide relates to that, but we see revascularization as well. But this is focused on cartilage and hair follicles. So, the cartilage here, this is an Alcium blue staining, so everything blue is cartilage. After day 35, you do not see much cartilage in the regenerated wound. But after about 100 days, you see very nearly full progression of cartilage formation within the wound. And we also see upregulation of chondrogenesis associated genes in the aftermath of the administration of the drug through that injectable hydrogel. We were very surprised by this, that in this large defect, we get basically a return to full density of hair follicles. So all of the hair reforms in this defect, and we can see these hair follicles by keratin staining. And also, we determined that the density is basically returned to the normal density of hair follicles. And you also see the upregulation of hair follicle associated genes after administration of the drug. About mechanism, and this is still, really a story that we're trying to unravel, but there seems to be the ability with this therapeutic to force cells into a progenitor cell state. I hesitate to use the word de-differentiate, but the in vitro experiments are shown here. Okay, again, I'm probably not the best expert on de-differentiation, but the evidence for progenitor cell marker levels on B6 cells exposed to this drug suggests that they are in a progenitor cell state. So the top row of these panels here are MRL fibroblasts. So you can see the high levels of HIF1 alpha in the fibroblasts from the MRL mouse, but other markers of progenitor cells, NANOG, OCT34, et cetera, indicators of progenitor cell status. Again, B6 cells, fibroblasts, isolated from B6 mice, normal mice, do not show high levels of any of these. But if you expose those cells to the drug, you get, again, high HIF1 alpha expression, and then basically recapitulation of the expression of these progenitor cell markers. And again, this is all dependent on HIF1 alpha because the siRNA experiment shows that these markers are inhibited with siRNA. So it's, again, pointing to HIF1 alpha as a key player in regeneration. And then these are the tissue sections from a similar experiment where we looked at a number of progenitor cell markers. So within the ear hole defect, you also see de-differentiation or progenitor cell status of some of the subpanel of these compared to the previous slide. Okay, so so these experiments up to this slide and published about 6 years ago, all utilized that gel system that I described about 10 minutes ago, and it determined, even though we designed that with hydrolyzable ester bonds, we determined that for reasons that we don't fully understand, that gel was very slow to degrade and actually ended up making defects, abscesses in the animals where we injected the material, and so we had to regroup and develop a new delivery system. And this was where my interest in supramolecular polymers really had an opportunity to make an impact. And so the concept that we derived was to exploit the drug, not just as the drug, but also to direct the formation of nanostructures within progenitor, a prodrug construct, a macromolecular prodrug construct. Okay, and we can do this because, as I mentioned before, the drug is very hydrophobic. So if you combine this hydrophobic drug with a hydrophilic polymer like PEG, in fact, we still use PEG, and then we do the conjugation of the drug directly to the PEG through this hydroxyl group here, a branched linker, and then conjugate the drug shown in blue to this polymer. We end up with polymers like you see on the left here. P73 is a linear methoxy on one end, so no drug on one end, and then 3 copies of the drug on the other end of the polymer. And then this is a 700 Dalton polymer, so low molecular weight PEG, and then a 8 kilodalton PEG, much higher molecular weight, with a total of 6 drug copies, 3 on each end. Okay, just focusing on P73. If you put this in water, okay, the whole system is designed for the blue part, the drug, which is hydrophobic, to drive assembly into nanostructures, and then through hydrolysis of these ester bonds here to actually release the drug. So it's a polymer prodrug that in vivo would hydrolyze to release that DPCA drug. We had evidence early that this was forming nanostructures through simple viscosity measurement. So if you put this polymer in water and measure the viscosity and do this in a concentration dependent manner, you see as you increase the viscosity, you get almost exponential growth at this beginning at about 1 or 0.5 milligrams per ml, evidence that there's some sort of nanostructure in there. And we also can see this by stochastic imaging. So here's an image of this P73 in water using a hydrophobic green dye. So the green dye is presumably in the core of the nanofibers. And you can see these are really, really long nanostructures here, and we ascertained that the viscosity increase here is due to entanglement of these fibers and increasing the viscosity of the system. We also did SAX, small angle X-ray scattering, to look at, and TEM to look at and confirm the presence of these nanofibers. So here's TEM, conventional TEM, and cryo-TEM. I'm not sure if you can see this, but there's fibrous structures in the cryo-TEM. And then the SAX also told us that we have nanostructures present, which are on the order of a few nanometers in diameter and consistent with all of the other observations by TEM and SUP, the viscosity as well as the imaging, stochastic imaging. And so what we think is going on when these fibers form is that you have partitioning of the drug, which is hydrophobic, into the core of the fiber. So the core of the fiber is composed almost entirely of drug, and then the PEG, which is linear, is emanating out on as a corona on these fibers. I'm a material scientist, so I like to do things like make phase diagrams. So this is a phase diagram of sorts of the mixtures of those 2 polymers. So the one that forms the fibers, which is this one here, P73, and mixed in different ratios with P80D6. So again, this is a much higher molecular weight PEG here, but 6 copies. And so the idea here was these would form fibers, okay, and this polymer here would cross-link the fibers, okay? And so we mapped out the gel and liquid phase diagram for various mixtures. Everything's shown in red here, sort of in this area here, and below the red curve here is a gel. And so the red line here above is the temperature at which these nanostructures melt. And so, you know, we're a gel in this space, and it's a liquid in this space above. All right. So there's two gel compositions in particular that I'll focus on, number 10 and number 12. These are sort of in the middle of the phase diagram, but actually at 37 degrees would be gels. Okay. And the idea here is that they can be injected into a wound site, and through high viscosity and viscoelastic properties would be retained within that location, and then release the drug locally. These systems have a significant amount of drug. So, at these two compositions, 10 and 12, the drug by dry weight is 18 to 20%. So reasonably good concentrations of drug. These mixtures are very interesting in a viscoelastic way. So these are a lower part of this is a rheology experiment showing the storage and loss moduli under shear. Okay, and so at each of these points here where we have a cliff, we induced high shear in the sample. So the prior to that, and the plateaus here, this would all be a gel. Okay, and then when you shear the system, it becomes a liquid. And again, we envision this system to be basically fibers that are tied together by these longer molecular weight molecules. This property of being shear thinning is very useful as an injectable gel. So we can load our gel into a syringe, and then when you press the plunger of the syringe, the high shear environment, as it's forced through the needle, the constriction of the needle, will liquefy the gel and make it injectable. And then when it comes out, it will reform a gel. And this is very rapid, as you can see, the formation of the gel. The drug release depends strongly on composition, and I don't have time to spend much here, except to say that we pay a lot of attention to the composition of the drug and try to achieve a release rate that is consistent with this 1 to 2 weeks observation of the need to upregulate HIF-1 alpha. So this is an example of the release of the number of the drug from the number 10 and 12 gels. It is between a week and 2 weeks of release of the drug. This looks remarkably similar to, this is an in vitro immunohistochemistry. It looks remarkably similar to a slide I showed you earlier, except this is now with the supramolecular gel instead of that previous gel. But the same story, you have MRL fibroblasts, which show the high levels of these markers. B6 fibroblasts, which do not express these markers, and then B6 fibroblasts exposed to the supramolecular gels, again, induced into a very similar phenotype compared to the MRL mice. And then these are the results of the animal experiment. So it's the same animal model now. And so we see upregulation of HIF-1 alpha, shown in green in the tissue slices, good ear hole closure, similar to the MRL mouse, and eventually, again, regeneration, early regeneration of the cartilage tissues and other tissues. We did not carry this experiment out to 100 days like before, so we didn't get full cartilage formation. So to summarize at this point, what we can say is that HIF-1 alpha is key to regeneration in the MRL mouse, and appears to be capable of, when upregulated through a PEG-D inhibitor, capable of inducing a epimorphic-like or MRL mouse-like regeneration program in an otherwise non-healing mouse. Okay, and I described what our current system is with this supramolecular polymer, where the drug is both structure-directing and therapeutic. And described the early results on this simple animal model, which is the ear hole punch, okay, where we get skin, cartilage, and hair follicle regeneration. I'd like to now talk about just spend the last 10 minutes or so talking about what's in progress, early results on things that are in progress, and I'll focus on periodontal regeneration and fetal membrane regeneration. So on the subject of periodontal regeneration, our goal here is to provide a novel treatment for periodontal disease. This is a progressive inflammatory disease that ultimately in advanced stages leads to resorption of the bone of the jaw, which is anchoring your teeth in. And this is due to bacteria that secrete factors which induce a very aggressive inflammatory response. This is a massive healthcare situation. So on the order of 60% of U.S. adults have, of over the age of 65, have moderate to severe periodontal disease. And 19 million U.S. adults have severe periodontal disease. The standard of care when you get to these moderate and severe periodontal disease is very aggressive, is actually a surgical procedure. Root planing and scaling, where you cut away the gums and then you scrape off all the bacteria and debris there. Antibiotic therapy, this generally is moderately successful, and if a disease progresses, then surgery with bone grafts and barriers and soft tissue implants are used. And actually, most of these therapies, including, the starting point, oral hygiene, are low compliance and poorly effective. Okay. And these surgical approaches are very expensive. Okay. And quite invasive. Okay. And so we would like to develop our approach for regeneration. The animal model is shown here. It's a simple ligation animal model. So you take a suture actually around and tie it around the base of a molar. Okay. And then you this is in a mouse, and then you let the animals put them back in their cage for 10 days or 2 weeks. And then you go back in, and what you observe when you go back in after a couple of weeks is aggressive resorption of the bone and advanced periodontal disease. So you can see that basically in these images here, where you have prior to the ligature, you see the little bit of the root of the molar, and then on this one here you can see very aggressive periodontal disease, which has developed quickly and led to resorption of the bone that's actually anchoring this tooth. Okay, so that's the animal model. So we would do this a couple weeks before administering our gel, and then see how the regrowth and regeneration of that bone occurs back in. So here's the animal experiment. So 3 administrations of the gel in the same way that I described earlier: day 0, day 5, and then day 10. This is with George Scarless at University of Pennsylvania, a periodontist. Here's some preliminary micro CT results. So this is the control animal, and again, you can see the exposure of the roots here due to resorption of the jaw. So this is the jaw down here. And normally it would be basically up at this level. The advanced periodontal disease has exposed the roots here through resorption of the bone. This is after 30 days of administration, and you see significant regeneration of the bone, almost back to the normal state. Okay. And this has just been published, actually is published late last year. And so this is, you can find this, and again, you see very significant gain in the bone amount in the defect site with our 1,4-DPCA. The amazing thing about this, compared to what is done now in periodontal treatment, is that we have not touched the oral cavity. We have not done anything inside of the oral cavity. We have injected subcutaneously, and we get regeneration peripherally. And that's basically a significant feature of what we've developed here. And this, again, is not an injection of a biomaterial into a defect. Okay, we are orchestrating this regeneration by administering a drug in the current situation here now is subcutaneously. Okay, it's all orchestrated from afar. The other one, let's see how I'm doing on time. So just a few more minutes on fetal surgery, regeneration in fetal surgery. So I've had a project on a repair of fetal membrane defects for quite a few years now. And this is a collaboration with Mike Harrison at UCSF. He was the first surgeon to actually ever do a surgical procedure on the fetus in the second trimester. And you know, these are rare, but commonly practiced in certain locations in the U.S. and throughout the world for these reasons that you see here, maybe a few others. The problem is you have to cut the amniotic sac to do any intervention on the fetus, and so that's where the problem lies, which is even by minimally invasive fetoscope intervention, you puncture the membrane, and this membrane is the amniotic sac, which holds in the amniotic fluid and keeps bacteria and infection out, and so it's a very critical wound. And the problem is that these often rupture after the intervention within a few weeks. Basically they rupture, and this gives rise to premature birth. Okay. Now the surgeons like to do these procedures between weeks 16 or 17 and about 25 or 26 in the shaded area here. If you look at survival of a fetus outside of the womb, it's basically zero before week 22 or so, and is low through this period of time. So if this procedure is done and induces premature labor and birth, it's quite a risk to the fetus. Okay, we would like to develop, and my project was to develop adhesives that can actually seal these membranes, and it actually does work in animal models and ex vivo models. So here's some published data showing we can actually improve survival rate in a rabbit study with a gel that actually seals the wound, improve the survival rate from 36% to 80%. Okay, so and that's been an interesting and fruitful project. But as we've progressed in this project, we've learned that the engineering and delivery in a clinically relevant manner to be able to do this is very, very challenging, and in fact it may not be possible to easily implement. And so we've started to think about, should we heal the membrane instead of seal it? Okay, and that's where we've come full circle here and started thinking about maybe taking the membrane and trying to heal it. And the conventional wisdom is that these membranes do not heal. Okay, so if you puncture or take a suture or take a scalpel and cut to do an intervention, that defect does not heal. And so we're starting to think about, can we use our approaches that I've described to upregulate HIF-1 alpha and maybe even get human fetal membrane defects to heal. And so we're just starting on this. So the end of the story is uncertain. But anyway, we can upregulate HIF-1 alpha in the amnion cells. We know that already from tissue level experiments, and also isolated cells, amnion cells from human membranes. And we can also get some early evidence in a dose response study with our drug of upregulation or a phenotypical expression of regenerator cell markers that may hint that we can get basically healing through this epimorphic regenerative pathway. Okay, so this is in progress, and I'll have to tell you about the outcome at another time. So I'll end here. And just add to my earlier summary that working on the jawbone regeneration in advanced periodontal disease, it's going very well, and very, very early stages of trying to pull this off in human fetal membranes. I'd like to thank my collaborators, particularly Ellen Hebra-Katz and George Hadjishengalis, Group members, particularly the data that I showed you, Jing Chang, Josef Strahin, Kelsey DeFreitas, Jisoo Shin, and Sally Winkler, and funding primarily from NIH for this project. Thank you again for the opportunity to give this presentation, and I am happy to take your questions.
Thank you for this fantastic talk. Many questions came in. So, does high shear at the time of injection destroy some of the cross links created by the high molecular weight polymer?
Yes, yes, that is actually a feature of the shear thinning. So the entire there's no covalent cross-linking in this gel. Okay, we get gel-like properties, elastic gel-like properties through non-covalent interactions, and basically those non-covalent interactions are strong hydrophobic interactions between the drug molecules that are conjugated to the PEGs, which are found in the core of the fibers. Okay, so under shear, we can pull one of those out and shear the system. Okay, and induce deformation. But that molecule is going to be hydrophobic, and it will go back into the core. So it's a dynamic bond, non-covalent bond, which we can disrupt, and then it reforms. And that's the basis for the shear thinning, and then the reestablishment of the elastic properties within a very short period of time.
Thanks. How is the degradation property of PEG for safety and toxicity consideration?
Yeah, so the system that I showed you relies on two different, the supramolecular system relies on two different molecular weights of PEG, 700 Daltons and 8,000 Daltons. Those would be within the molecular weight range that can be eliminated through the kidneys. So we have not seen any injection site toxicity of the material. And so we think the biological profile, biocompatibility is pretty good for the injection site.
Thanks. You mentioned efficiency of healing of a 2 millimeter symmetrical puncture. Have you measured the efficacy of healing of an asymmetrical wound under your system?
Yeah, it's a good question. We're starting to do that in these other experiments, which I described that are ongoing. So we haven't done this in the ear hole punch model and done a asymmetrical wound. Would be interesting to do that, but we've progressed to more advanced models for creating the wounds, and in the periodontal defect, it's quite complicated.
Thanks. While there are several other molecules for PHD inhibition, I'm wondering the potential behind using 1,4-DPCA despite its poor solubility, as you have kindly elaborated.
Yeah, that's a very good question. There are a number of other PHD inhibitors. We have done some comparisons of PHD inhibitors that are approved, and for example, the Fibrogen one, and we've found that they do not have the same ability to induce regeneration as 1,4-DPCA. We can't fully attribute that to anything yet, but it probably has to do with which of those PHDs are inhibited, and this drug that we're working with may have some affinity for inhibiting, a different PHD than the others. So we're still trying to understand that, but the initial observation is that the other PHD inhibitors that we've looked at do not, they upregulate HIF, but they have not produced regeneration in the same way.
Thank you. Another one is PEG-DPCA itself capable of stabilizing HIF1 alpha without cleaving from nanostructures?
Another great question. We have early evidence that actually that the low molecular weight one can actually, before hydrolysis of the drug, release of the drug, it can actually inhibit or upregulate HIF-1 alpha. That's a preliminary observation. So I'm not gonna hang my hat on that yet, but there is an early observation that before it's hydrolyzed, that construct actually can get into the active site and inhibit PHD. Presumably it's inhibiting PHD. And so we have to do more to confirm that, but it would be an interesting observation because we've always thought of the drug conjugated to the PEG as being an inactive prodrug, and it activates the drug only by hydrolysis and release. But we are building some evidence that actually it's more complicated than that.
Thank you. In your study with HIF-1 alpha, did you see cases of cancer formation when you upregulated its level?
Wow, these are all great questions. Yes, we get this cancer question a lot, and the basis for that question, I believe, is that there is an association between the high levels of HIF-1 alpha and solid tumor development, and probably because in solid tumors the core is hypoxic. And so there is concern about HIF-1 alpha, and upregulating HIF-1 alpha, are you going to increase the risk of cancer or metastasis? We don't know the answer to that question yet, but we can say the following. One is that MRL mice do not develop cancers at higher rates. They have high levels of HIF-1 alpha and do not develop cancers at higher rates than other mice. And also, our drug delivery regime is very limited. So we're delivering drug for a couple weeks, 2 weeks, roughly. That's our goal. And then there's no more upregulation of HIF-1 alpha. This is all part of the MRL program. It's a very transient upregulation. So if that works out and plays out to be a therapy, there's no long-term upregulation of HIF-1 alpha. But this is a good question, and it's something we have to pay attention to.
And one final question. Do you also observe a metabolic shift from oxidative phosphorylation to glycolysis upon HIF-1 alpha stabilization with PHD inhibitors? Are there any connections between the two pathways that support your regeneration model?
Yeah, we have not done that experiment. That's a really great comment as well. So I unfortunately I do not have the answer, but it's a very good point.
One final question. Thank you. Have you considered other materials besides PEG for your materials, and perhaps natural-based hydrogels as a synthetic graft, as a graft of PLA? Or
yeah, we're beginning to do some stuff there. So PEG is not probably going to be the solution for everything, and so we're beginning to think about other systems that do not involve PEG. But it's too early for me to really elaborate on that because it's in the early stages. But the quick answer is yes, we're looking at other approaches that do not involve PEG.
What are your thoughts on neuronal tissue regeneration?
Mm-hmm. Yeah, we have. Well, Ellen has actually done nerve specific staining of tissue sections and finds evidence for regeneration of nerves. And so there's that. I don't think that's the full story yet, but there is evidence of nerve tissue regeneration.
Excellent, excellent. Well, thank you for this fantastic talk. I appreciate your time, Phil.
Thank you very much. I had a great time. So if you have any questions, just let me know.
All right, thank you. Have a nice day.
Great. Thank you.